Oh boy. So we've learned a little bit about DNA and the structure of DNA and how it can be copied or replicated. We've learned that DNA is an incredibly important molecule for living things, that it acts as the hereditary information that's passed on from cell to cell, from parent to offspring. And we've also mentioned that it is the genetic information in organisms. And today we're going to learn about what that means. How does DNA fulfill this role as the genetic information for organisms? That information is, in fact, the information for making proteins. So, how a living thing functions is mainly determined by the proteins it builds. Proteins make up lots and lots of important structural molecules in cells. Proteins make up lots of important transport molecules that determine what enters and leaves cells. And proteins are the category of organic molecules that enzymes belong to. And enzymes do the chemistry inside of living cells. They perform the chemical reactions that separate the living world from the non-living world. So proteins are enormously important to living things, and the genes in our chromosomes are instructions for making proteins. Mostly. There's some exceptions to that, but mostly that's what genes are for. And really, you can consider each gene in our DNA to be like a recipe for making a particular protein, a set of instructions for building that protein. And that recipe is written out in sort of a coded language in the bases of DNA. Those A's, T's, G's, and C's in a DNA molecule will determine the sequence of amino acids in a protein. And that sequence of amino acids will determine the structure and the function of the protein. So it's a big picture there, and please pause the video whenever you need to write down some notes, to write down some questions, to look something up. We're going to go through all of this in some detail in the next few minutes. So DNA is the genetic information, the information we need to build proteins. But there's a problem. DNA molecules are stored in the nucleus for good reason. Keep them safe in the nucleus, keep them away from enzymes and other chemicals out in the cytoplasm that might damage the DNA. We've got our recipe book for being a living thing, for being a human in our case, stored in the nucleus. We want to keep those instructions, those chromosomes, safe in the nucleus. But the machinery, the factories for making proteins are out in the cytoplasm. Those are the ribosomes. So how do we get the instructions for making proteins from the nucleus to the ribosomes? And the answer is in another kind of nucleic acid that's called RNA. And we will take a look at RNA and put these pieces together for how DNA and RNA work together to help get us our proteins made out there on the ribosomes. So RNA is a nucleic acid, like DNA. RNA has a lot in common with DNA. It has, for each nucleotide, a phosphate group, a sugar group, and a nitrogenous base. Here's our DNA over here that's double-stranded, phosphate, sugar, nitrogenous base. The phosphate is exactly the same in both DNA and RNA. The sugar is slightly different. And if you look at the ribose sugar, that is the sugar unit in RNA, and compare it with the deoxyribose sugar, that's the sugar in DNA, you can see the highlighted difference. There's an extra oxygen atom on ribose compared to deoxyribose, hence the name. And then the other major differences between the two molecules are in the 4 bases that build DNA, A, T, G, and C. 3 of the 4 are the same for RNA. The adenine, guanine, and cytosine are exactly the same, but RNA does not have thymine, T. Instead, RNA nucleotides have a very similar base called uracil, and we'll look at that a little bit later. But those 2 bases chemically are very similar and behave almost identically in terms of how they function in a cell. And then the final difference is that RNA tends to be a single-stranded molecule rather than the double helix structure of DNA. Now, it's possible that RNAs can do some hydrogen bonding because A's, G's, U's, and C's can be complementary to each other in the same way that DNA nucleotides are. But for the most part, RNAs are single-stranded. And for sure, they don't form the double helix that we see with DNA. So RNA, lots of similarities with DNA, but some important differences. So 3 different types of RNA are involved in this process of converting the information in a gene, in DNA, into a protein made out on a ribosome. And those 3 types we'll talk about individually. They are messenger RNA, ribosomal RNA, and transfer RNA. Each one plays a critical role in the production of a protein. So, messenger RNA first. Messenger RNA, or mRNA, is essentially an RNA copy of the instructions for making a protein in a gene. So we've got DNA in the nucleus, a sequence of bases on DNA. Messenger RNA has the job of carrying that sequence information away from the nucleus and out to the cytoplasm. Ribosomes are made of RNA and proteins. And this rather complicated looking model that's pictured on this slide is depicting the large subunit of a ribosome. Ribosomes have 2 parts, a large and a small subunit. And in the large subunit of a ribosome, the structure is dominated by protein, which is purple in this diagram, and RNA molecules. And there are several different kinds of RNA. You can see orange, white, red, green, and some sort of purplish up here, maroon, reddish purple. Anyway, all of those are RNAs, and just the lighter colored purple is protein. And ribosomes by mass are pretty close to about 50-50 protein and RNA. So, second category of RNA, ribosomal RNA, or rRNA, is a structural part of the ribosomes that make proteins. Third category of RNAs, super important, crazy cool, transfer RNAs, tRNAs. And tRNAs are responsible for delivering the amino acids to the site where proteins are being built. Recall that a protein is made of amino acids. A protein is a polymer made of amino acid monomers joined in a chain. So to make a protein, you've got to bring a sequence of amino acids together and put them together, to bind them together. Well, the carriers for the amino acids, the little shuttle buses that deliver the amino acids to a ribosome to make a protein, are the tRNA molecules. And you can see 3 different versions of a tRNA model, and these are all of the same tRNA. And depending on what your interest is in biochemistry, one model might be more appropriate than the other. The sort of cloverleaf model, as the one on the left is described, are the ones that you see most commonly. Amino acid on the top of this diagram, and the very important part of the tRNA called the anticodon is on the bottom, and we'll get back to that in just a bit. So I think the last slide for this video, again, just coming back to a big picture about what's going on with converting the information in a gene, the information in a segment of a chromosome, they're in our nucleus. How is that information used to build a protein? And so the 2 big steps for building a protein occur first in the nucleus and then in the cytoplasm at a ribosome. And transcription comes first. This is where the information in a gene, the sequence of bases on a segment of DNA, in other words, those are converted into the sequence of bases on an RNA molecule. And that sequence is the critical information. And that sequence will be used to determine the sequence of amino acids in a protein, which determines what protein it is. And if you look at the diagram here, you can see up here on the DNA at the top the sequence of DNA nucleotides T, A, C, C, G, etc. And if we look down at the single-stranded messenger RNA that was made by this process of transcription, we can note that the nucleotide sequence on the RNA is complementary to the nucleotide sequence on the DNA. And in essence, we can read the sequence of the mRNA and know exactly what the sequence of the DNA is. So that information from the DNA is transcribed into RNA form. So A, adenine, is complementary to T, thymine. U, uracil, is complementary to adenine. Guanine, G, complementary to cytosine. Guanine, cytosine. C, G, U, A, G, C. So the sequence of DNA has been preserved in this sequence of messenger RNA. Doesn't look the same, but the information is there. And that's transcription. Translation happens out on the ribosomes, either free-floating ribosomes in the cytoplasm or those ribosomes attached to the rough ER. In either case, we are outside of the nucleus now, out in the cytoplasmic milieu when translation takes place. And that is the part of the process where the sequence of the nucleotides, in this case from the mRNA, are used to determine the sequence of amino acids to build a particular protein. And in this case, we call this translation because it's kind of like we're changing languages completely. We're going from the language of nucleic acids to the language of proteins. And that's translation.